Thank you very much for coming in the room as well as online. It is my pleasure to introduce Chris Fuss. He is a forest ecologist and an associate professor here at the university. And he'll be talking about alternative stable states in New Zealand forests.
Thank you, Josh. Well, good afternoon, everybody. Today I'm going to tell you about an experiment that makes up one of 4 strands in my Martin project that's just coming to which looks at landscape partitioning between different mycorrhizal associations in the South Temperate Zone. And that's really Nothofagus versus the rest, because at least in Chile and New Zealand, Nothofagus is an ectomycorrhizal outlier in loner in tree floras that are dominated almost completely by arbuscular. I'm going to start this story by taking you briefly away from the South Temperate Zone to tropics. Yes. Here we are. So, tropical moist forests are, of course, a byword for diversity, as you can see quite well here in this photo. You can easily find 150 tree species per hectare in moist tropical moist forests. So imagine what a shock you'd get if you're walking through a tropical forest and suddenly you walk into a stand and one tree is completely dominant, because that occasionally does happen. Here is a monodominant stand of this tree in the walnut family in montane forest in Canada. So this species, Orimonea mexicana, dominates all tiers of the forest, the understory, subcanopy, and the canopy, which suggests that it's a stable, compositionally stable stand. It's not an early successional stand that's going to be invaded by other species. It seems to have a pretty stable hold on these sites. But another example from tropical Africa, this is a true equatorial rainforest right on the equator in Congo, and this is an even more overwhelming example of monodominance. I'm not even going to try and pronounce that name, but it's in the pea family, and from the seedlings right up to the forest canopy, the species is overwhelmingly dominant. So monodominant is commonly, it's kind of a convention to rule of thumb that greater than 60% dominance, well, that monodominance. But that last example we just saw is much higher than 60%, more like 90%. And most of these tropical monodominant stands appear stable. Not all of them, but most of them appear stable. And the boundaries are not easily explained by topography or soil type. So if this white patch here represents a monodominant stand here, it's not like the soil is. It's different under here or inherently different from over here. It's not like these are on ridges, topographically differentiated. So we seem to be dealing with alternative stable states with these monodominant stands. Now what's going on here? This is first noted, I think in the 90s, people started commenting on this. And there's been several theories about what's going on, what's driving these monodominant stands. Well, what these 2 trees that I've just shown you have in common is that they're both ectomycorrhizal. Now, this turns out to be true of most, but not all, of the trees that form these monodominant stands in tropical moist forests. So, why is this important? I have to do some brief refresh here, I think, about mycorrhizal associations. I'll only talk about 2 types here, the 2 most widespread types. Most terrestrial plant species, that figure is probably a bit too high, but most form arbuscular mycorrhizas with those fungal groups. And that includes most tropical moist forest trees. The main benefit of arbuscular mycorrhizal infection seems to be enhanced access to greatly increases the absorptive surface of the plant. So there's enhanced uptake of nutrients, mineral nutrients like phosphate that don't diffuse very far in the soil. The other well-known type is ectomycorrhizas. These are common in the North Temperate Zone and in the boreal forest. Most trees are ectomycorrhizal. Different fungal groups from arbuscular mycorrhizas, and there's a small minority of tropical moist forest trees that are ectomycorrhizal. Now, some ectomycorrhizal fungi, they have a superpower that I'm going to, that some of you may know about, but I'm about to explain. They can short-circuit the local nitrogen cycle in a way that gives their host trees privileged access to nitrogen at the expense of everybody else. Here is a couple of slides contrasting local nutrients, local nitrogen cycling in arbuscular versus ectomycorrhizal vegetation. So what you can see here is what you'll see in most textbooks written before, well, until very recently, as normal local nitrogen cycling, right? So you've got the decomposers breaking down the organic matter, and that decomposition process eventually results in the liberation of ammonium and nitrate soil solution, so you've got mineralization there. And that mineral nitrogen is taken up by tree roots and by arbuscular mycorrhizal. So the decomposers and the arbuscular mycorrhizal fungi are doing quite different things. Quite different things. Things get a bit blurred in ectomycorrhizal vegetation, where you can get an organic nitrogen economy, as it's been termed. Some of these ectomycorrhizal fungi, they retain part of the enzymatic repertoire of their decomposer ancestors. So they can extract organic nitrogen from dead organic matter, cutting out the middleman, so to speak. That's the decomposers. So I've attempted to attach some ectomycorrhizal hyphae there that are going into that dead organic matter and kind of preempting the decomposer, or at least competing with the decomposers. So well, funnily enough, the decomposers are not terribly happy about this. They've got competition for nitrogen. They have to deal with a substrate that's been impoverished in nitrogen, and so decomposition slows down and you get less mineralization. So this is how this short circuit works. And you can get at least in some circumstances a positive feedback setup that perpetuates ectomycorrhizal dominance on the site. It's abundant ectomycorrhizal trees, abundant fungi in symbiosis with them. Fungi compete with the decomposers for nitrogen, which slows down decomposition and mineralization. So arbuscular mycorrhizal plants that don't have this privileged access to nitrogen can be starved of nitrogen. The supply of nitrogen is greatly reduced, so their survival is reduced. So most opportunities are captured by seedlings of ectomycorrhizal trees. That's the positive feedback that's been demonstrated pretty comprehensively in this tropical forest with the Oreomonea mexicana, the walnut family tree. So it's quite likely that Nothofagus, which is also ectomycorrhizal, is doing this sort of thing in New Zealand. Sometimes if you go into a beech forest like this one, you can see how the understory is quite sparse. In fact, the forest is quite open. That's not just the deer. The deer are contributing to the pattern here, I would suspect, but the understory the forest is quite open. It's quite a simple structure, and there's just a kind of narrow range of species in the understory. So Briefly, I'll talk about Nothofagus distribution in New Zealand and how landscapes are partitioned between Nothofagus and other tree species. So Nothofagus, of course, is, as some of you at least will know, it's mainly in colder parts of the country, most abundant down south and at higher elevations. In the lowland North Island, it's an interesting pattern that there's often a topographic segregation. So it's often on ridges and spurs. I've got a slide here. This is a satellite photo of hill country in the Rangitike Wanganui area. And these dark smudges that you can see here, these dark streaks, those are Nothofagus ciliandri stands, black beech stands on spurs and ridges. And the brighter green is conifer-broadleaf forest, which is occupying most of the faces of most of the gullies. So there's a topographic sorting or segregation here. But even though there is this definitely sorting and segregation across landscapes at different scales, there's a lot of overlap between the environmental tolerances of Nothofagus species and arbuscular mycorrhizal trees. And so you can get situations where, of course, where you get mixtures, and what's more, you can get, in some places, occasionally these monodominant Nothofagus stands within a mosaic of more diverse forest. Anybody been, this is Rohini Corner, anybody been there? No. Okay. So it's, I'd seen a paper about this stand, and it was a fascinating read. And I was walking across this huge clearing, and I looked up and thought, oh, there it is. You can just see the pattern quite obviously there as you approach the hut. These dark patches here are monodominant Nothofagus ciliandri stands. They're almost pure Nothofagus ciliandri in a matrix of more diverse conifer-broadleaf forest. The biggest trees are Libocedrus, that's Kaikawaka, and pig pine, Pelecarpus biformis. So this looks kind of eerily familiar. We think of the tropical example of monodominance. Here's another parent example, which I've been so with the experiment I'm going to show you was done in the eastern Waikato Upper. So this is actually a covenanted forest on private land, and it's mostly conifer-broadleaf forest, but there are these Nothofagus stands here, these darker patches once again, which are Nothofagus stands. Now, at this site, there is a degree of sorting. So the Nothofagus tends to be on poorer soils than the conifer-broadleaf stands, but there's overlap. So here are some, I went around and drilled lots of holes in the ground and sent lots of soil samples off to the laboratories to try and find stands that were on, of Nothofagus and conifer-broadleaf that were on very similar soils. And eventually, after spending quite a lot of money, I eventually found these stands. So there's actually a lot of overlap in total nitrogen. If you look on here on this left-hand graph, so there's it's pretty easy to find stands that have same nitrogen concentration. Not quite so easy with phosphorus, but you can compare stands that are very similar in terms of total phosphorus. So I found these 3 beech stands here, identified those 3 beech stands, and 3 conifer-broadleaf stands with very similar total soil phosphorus and nitrogen and similar topography. They're all on north-facing slopes or ridges. I should just mention that these two fine texture patches here are Carnuka stands. They're post-fire stands, that kind of a third element in the landscape here. Stands that have been burnt perhaps 50 years ago. So we used these 6 stands for our experiment. So a quick look at these 2 forest types. The conifer-broadleaf forest at Rocky Hill is it's pretty different from what you are used to seeing around here in the Waikato district. It's cooler and it's drier, and it's also, I think the composition has also probably been altered by logging and fire to some extent, but these stands you can see here certainly haven't been logged or burnt for at least 150 years, maybe 200 years. And Hino is a dominant canopy tree, which is pretty unusual. Pigeonwood is also very common. A lot of silver fern. Polycarps are actually quite scarce. They're scattered at low densities. That might reflect history, but they are scattered throughout. The beech forest is, as you can see, looks very different. Over 90% of the basal area is Nothofagus cylindri. The understory is more diverse, but very interestingly, you've got, as well as the Nothofagus saplings that are indicative of self-replacement in situ, there is a lot of Ericaceae in the understory. Now, the Ericaceae they're another group of plants that have a third type of mycorrhiza, the ericoid mycorrhizas, that also give access to organic nitrogen. They can do a similar trick to the ectomycorrhizal fungi. So they're quite at home in this environment. They can prosper in this environment in a way that most arbuscular mycorrhizal plants can't. Can you see a tree fern in this photo here? I can't. There's almost no tree ferns in the beech forest, whereas they're common in the arbuscular mycorrhizal forest. Now, there's an obvious difference in the structure of these forests is the canopy structure. The canopy is much leafier and denser in the conifer-broadleaf stands, so the understories are much darker. There's only about half as much light in understories on average as in the beech forest. And this is something you can see when you go tramping. I know I've heard people who are kind of don't study forest, for example, say, oh, beech forest is so bright and I feel more cheerful than the beech forest because there's more light coming. And that's because of differences in leaf area index. It's at least 1 unit of difference there. So this is the average number of leaves that an imaginary beam would have to pass through to reach the forest. So we hypothesized that we have positive feedback stabilizing both these forest types. So we've got the familiar on the left there in the beech forest. We've got that same kind of organic nitrogen economy that we saw in that tropical monodominant forest. And the beech forest, what we're thinking is that the understory is simply too dark. Sorry, the broadleaf forest. The understory is simply too dark for beech seedlings, because the Fuscospora group Nothofagus species are probably the most light demanding tall trees in the country. And this is borne out by low light survival data that have been published by the land care people. They've support the impression that these beeches are the most light demanding trees in New Zealand. Late successional tree flora. So beeches just shut out by lack of light. So we set out to test both these hypotheses. This is here I'm showing how I think shade keeps Nothofagus out of conifer-broadleaf stands. So we would expect then the beech species to have a higher light compensation point of growth than any of the species from the conifer-broadleaf forest. So this is light availability here on a log scale, and here above ground relative growth rate. So the compensation point is where net growth is 0, so it should be higher in beech than in any of the species from the conifer. And if we've got an organic nitrogen economy hurting the arbuscular mycorrhizal species in beech stands, and we should have something like this going. That the growth of the AM species, that's the conifer-broadleaf species, should be depressed right across all the light environments because they're just struggling to get enough nitrogen. So they probably have to allocate more biomass below ground to make ends meet. Whereas beech should be kind of indifferent, it shouldn't really mind which forest type it's growing. We grew 5 species from the conifer-broadleaf forest. Turned out some of these were good choices and some of them weren't quite so good. So 4 of these are arbuscular mycorrhizal. The one in the middle, rewa, it's like most Proteaceae, it's non-mycorrhizal. And that means that, like the arbuscular mycorrhizal species, it should not have access to organic nitrogen. So it should behave much like we expect it to behave like the arbuscular species. So let's see the beech seedlings. There they are, black beech seedlings that we grew and planted. Now, as some of you may know, you can't just go and plant seedlings in the forest in New Zealand and expect them to be still there when you come back for your quarterly visits, unless you protect them from the introduced menagerie. So this is a technology. This is kind of cool. It's a technology we've used before on my previous mats, and it works well. Whack in these 2.4 meter white posts, whack them in, and then wrap this tough. We thought it was pig-proof polypropylene mesh around these, but that's not enough. You've got to put pest spikes around the top to keep possum out. Because if possums can get anywhere near your experiment, they will mess it. And this works well. This setup works well. It only failed once. We had a pig busted its way into one of these plots. It wasn't clear why, because once it got in, it didn't seem to do much. But it made a determined effort, forced its way under mesh here, snapped the cable ties went flying, and then it sort of dug at the ground a little bit and then just disappeared. Unfortunately, they left possums in and nipped the tops off. We lost some data points there. So we used this wonderful instrument, the Lycor plant canopy analyzer, to measure light environments above each seedling. This is a terrific instrument that gives you high quality data. So here we're just showing you roughly the distribution of the plots. We cited the plots. Now these are not randomly cited. There wouldn't be much point at that. They're cited to maximize the range of light environments, grow species across the wide range. So some beneath tree-fall gaps, some beneath the darkest parts of the stand, some in intermediate environments. There's 6 plots in each of the beech stands and 6 plots in each of the broadleaf stands. I haven't mentioned these. Now these, Ian Dickey, my collaborator Ian Dickey, suggested these, and we've got mixed results with these, but they seem to have worked. They seem to have given us very believable and very conclusive data in this experiment. These plant root simulated probes, they have an ion exchange membrane there that adsorbs ions from soil solution, one for cations, one for anions. And we did two rounds of burial. We buried in the spring when thought that mineralization might be limited by temperature, and in the autumn when we thought it might be limited by soil moisture. I'll show you those results. I think they're the first results actually, and yeah, they're actually the most conclusive results. There is no nitrate under the beech. There is no nitrate under the beech stands, or practically no nitrate. You don't really need stats for results. It's so unusual in ecology to get a result that's conclusive. It's like, so pretty clear evidence that the ectomycorrhizal fungi are crashing mineralization. Louis Shipper's listing, yes, he is. He had something to say about this as well, I might mention shortly. Ammonium. Now the ammonium pattern is a bit different. We found no difference. There's less ammonium than nitrate, but no difference between the two forest types in either season. The almost complete lack of nitrate means that ammonium is the dominant form under the beech stands. And Louis Shipper said that wondered if there was either the trees or the fungi were also inhibiting nitrification. Interesting idea that we might look into. So as well as short-circuiting the nitrogen cycle, maybe there's some nitrification inhibition going on. No effect on phosphate. So it doesn't seem to be evidence of an organic phosphorus here. It's really just a nitrogen. Similar concentrations beneath the two forest types. I'm going to show you one more ion. Might be a surprise. That's manganese. Why am I showing you manganese? Because ectomycorrhizal fungi, some of them at least, produce manganese peroxidase, which is one of the enzymes that's involved, a key enzyme involved in breaking down inorganic matter and getting the nitrogen out. So sometimes you find elevated concentrations of manganese ions under ectomycorrhizal vegetation, and we seem to have it here, particularly in the autumn. We've got evidence of quite a lot of manganese in the soil solution. So this is further evidence of an organic nitrogen economy. We take that as further evidence of organic nitrogen. So that's the soil nutrient data. Now I'm going to show you some data on how the seedlings responded to the different growth environments. And so there's a graph showing the growth of each species in relation to light. Okay? Note that the light is shown on a log scale because plant growth doesn't respond linearly to light, it responds to log light. And light on an arithmetic scale. So each point on the graphs is the mean of the two plants on the plot in cases where the two plants survive, because in a lot of cases they didn't, like pigs, big growth, etc. Yeah, but some plants just died for reasons unknown. So that each point, each dot sometimes represents one plant, sometimes two, but the two plants are on the same plot. Now, that's a more ecological sort of result, I'm afraid to say. It's a more typical ecological result. So yeah, I'm too embarrassed to show you the R squared of the growth. It's pretty close to a round data globe. Now, I've got a comment about the lines on the graph. What I'm showing you there, those lines are not ordinary least squares regression, they're major axis. Now, I'm showing you major axis because we're not interested here so much in predicting y from x. We're interested in comparing the slope and elevation of the two lines. To do that, you need major axis, which is the first axis of the PCA. That's exactly what it is. And so we're asking if, in each case, if the species in question grows slower in the beech forest, and if there's an interaction there. So if there's an interaction, there'll be a significant difference in slope. So I'll show you the stats. Hino was a species that didn't behave very well. This is a species that it didn't. My impression is that perhaps it's not a great species for transplanting. That some individuals grew well, but look at this plant down here. We gave it everything. And it just sat there and sold. It's got like 7% light. So some of them took off and some of them just sold. So we don't really have a lot of statistical power, say, to detect differences there, but we just can't. We have no evidence of a difference between the two forest environments, which goes against the hypothesis. Pigeonwood is a better behaved species. We do have a difference in elevation. On average, it grows more slowly in the beech forest. So that's consistent with the hypothesis. It's an arbuscular mycorrhizal species that's struggling apparently in the beech forest. Rewa. Yeah, this is another poorly behaved species. So we've got a hint of a almost significant difference in slope there. What's going on there? Rewa, as it's a non-mycorrhizal species, we expected it to grow slow in the beech forest, but we don't have any evidence of that. It doesn't support the hypothesis. Lemonwood, now here at last, is Lemonwood is a model species. So it was very unhappy in the beech forest, as you can see. A big difference in the growth rate between the two forest types. And look at the tight grouping of the plants of the points in the arbuscular mycorrhizal forest and the conifer-broadleaf forest. Great species that transplants well. I can recommend it. Tortora, another species that I can recommend, is a good one to work with. So that's another nice R squared for at least in the conifer-broadleaf forest. We've got, and again, a significant difference in elevation, which means that Tortora grows more slowly in the beech forest. Finally, black beech, which is another badly behaved species, I'm afraid. It's problem here is we got seedlings from two provenances, well, part of the problem at least. We got seedlings from two provenances. We raised some ourselves from Rocky Hill Road. I bought some from a nursery, and the nursery seedlings weren't good. They some of them just sort of started dropping leaves into the ground. So we actually had to replace some of them. So the beech data are not great. Anyway, we've got a hint of a difference in slope, but it's not quite significant, and a hint of a difference in elevation. I don't know why beech would be unhappier growing in the beech forest than in the conifer-broadleaf forest, but we did notice that they got more defoliated in the beech forest. That there was damage from chewing insects on the seedlings in both forests, but leaf miners were present on almost every seedling in the beech forest. So maybe there's an effect there of natural enemies that they escape to some extent if they're planted outside. I didn't see that coming. Now, compensation point data. Here we're trying to find out if beech is the most light demanding tree, and if its compensation point is basically too high for it to grow in the conifer-broadleaf understories. Now, unfortunately, some of these data are, well, you can see the confidence intervals. Yeah. There's only two species that have sort of useful confidence intervals, lemonwood and Tortora, right? So we can have some confidence in those estimates of the compensation points. The others, because the relationships between light and growth are so weak, I don't think we can say much at all about the compensation points. And there are no significant differences here in compensation points. Even if beech has got the highest, it doesn't differ significantly from, I don't think, from any of them. And what's more, its compensation point there, according to this, is about 1% light, but it's the average understory light environment in the conifer-broadleaf stands is about 2%. So it's not clear why it's not establishing in conifer-broadleaf stands. So we don't know why it's not invading conifer-broadleaf stands. So to sum up, we do have evidence, quite good evidence to support one of our hypotheses about feedback mechanisms. We've got evidence of that short-circuiting of the nitrogen cycle stabilizes Nothofagus dominance at Rocky Hill. So we've got nutrient availability data showing an organic nitrogen economy, and we've got suppression of growth of some, but not all, arbuscular mycorrhizal species seedlings in the Nothofagus stands. The other hypothesis, that's not so well supported. We do have evidence that the conifer-broadleaf stands are much darker than the beech stands, but they're possibly not too dark for Nothofagus seedlings, and our compensation point data are frankly inconclusive. I'm not sure what we're going to do with them. And that is that. Acknowledgements, of course, to the Royal Society of New Zealand and to Ian Dickey, my main collaborator on this project. And Forest Enterprises are a great firm to work with. They own this covenanted forest. it's a whole lot easier than working on dockland. As some of you will know, you've had parallel experiences, and a legion of helpers who've helped in various ways in the field, or seedlings, or being field contact people, that kind of thing. So yes, thank you. And we've got a few minutes for questions. Yeah.
Good question. These stands, how do these, what's your hypothesis on how they get started? You thinking priority effects that they establish, and then these mechanisms keep them within that stable state, perpetuate the sort of pure stand of beach versus the mixed buttercup forest?
Yeah, presumably it's some kind of disturbance event that flips that, and you get a disturbance event, and then that coincides with a set of circumstances that favour one type of forest or the other. So it's a disturbance, and it coincides with a beech mast year, and so beech seedlings everywhere, and would strike at beech seedlings. You'll get, of course, it won't be pure beech initially, but if it's kind of within the environmental envelope where you get an organic nitrogen economy, then beech eventually takes over completely. And conversely, if perhaps a beech stand burns or is destroyed by some other kind of disturbance, or maybe there's a dieback, something like that, an attack of pinhole borer or something like that, and if that lines up with mast seeding by Hino, whatever, then you could get disturbance, I think, of some kind would be what flips the switch.
So I think about some windthrow stands around Cass beech forest there, and the density of seedlings coming through a beech, are just incredibly thick. So really probably out competing anything else to get established. So yeah, I mean, I don't think that's exclusive to the mechanisms you're proposing. There's obviously something here with the amount of the seed bank.
So what elevation are we talking about at Cass?
Well, I'd say that's probably definitely above 700 metres of sea level, something around 800.
That's probably within, there's a bunch of other nature trees that could grow there.
Yeah.
Beech has got hold of that site over quite an extensive area. But you could, there's no reason why I imagine, say, thin-bark telstra couldn't grow there, or broadleaf, but beech perhaps is shutting them out. Charlie had his hand up a while ago.
Okay.
Who's actually doing the questions? Me or you?
You guys the questions.
Yeah.
My question is, do you know what nitrogen species, organic nitrogen species, are being transported by the mycorrhizae from the? So, which organic compounds do they? You don't know, because to me, if they go for the obvious option, which is glutamate, that explains why you don't have nitrate, right? Because the decomposition of glutamate leads to nitrate. And glutamate is also the primary thing, the primary organic nitrogen species that's transported within the plant. So and I think there must be literature on what the ectomycorrhizal do.
Okay. Yep, I'll talk to Ian about that. Yeah, right. Thank you. Yeah, so Louis' nitrification inhibition hypothesis might be superfluous. Right.
Internationally, what's the explanation for dominance of AN species? So is it ecto species always seen as tending to be more light demanding? Or is there some other explanation? So that's just a New Zealand.
Yeah, we do. It's New Zealand is, I mean, the South Temperate Zone, we've got this, it's very different from the North Temperate Zone, where of course you have a whole bunch of different ectomycorrhizal genera, including very shade tolerant genera like fagus, like beech, and like hemlock.
But different herbs.
Yeah, I don't know. They're shade tolerant.
Well, there's a huge range of them.
Yeah, no, my pair of hypotheses doesn't work for ectomycorrhizal forests in general. I've suggested it works here in this situation because we've got, what's that face me?
So I'm just wondering why. So why is it that you have these vast AN forests that the Or, you know, what's the explanation for the stable state of the AN?
Right, it's in the tropics.
Yeah.
Okay, so arbuscular mycorrhizal fungi are believed to cost the plant less. They're the cheapest option. Yeah. So in environments where nitrogen is not too limiting, then they're probably the more cost-effective option.
Well, that would raise the compensation pool. If you've got a more expensive association, then you're going to need more energy to.
In the ectomycorrhizal species.
Yeah.
Yes, although that doesn't stop, say, hemlock or beech being very shady.
That manganese result. So, I mean, you're saying it's a byproduct of nitrification process or something that's why it's more likely to be detected in your probes.
A byproduct of the extraction of organic nitrogen from dead organic matter by the ectomycorrhizal fungi.
I'm wondering if manganese is more available. Well, yeah, I mean, I guess it's how the probe works and what form of manganese is that's detected in the probe. You wouldn't want to have a situation where the site actually fundamentally is different, right? Like manganese is prevalent. That, you know, it is routed through some biotic process. So
We should get total manganese from the soil and see if that.
yeah, I just
Okay.
wondered about
Yeah,
that.
we could do that.
Because I was thinking, you know, if these are sort of like disturbed sites or something, it might be fundamentally something different about the soil.
Yeah.
Biochemistry, the chemistry.
I'll talk to Ian about that. Yeah.
Don't know.
So I should put in just a bit further about that question. Not all ectomycorrhizal fungi do this hacking of the nitrogen side. Not all of them. They're the plant partners with them when nitrogen is hard to get. Nitrogen is hard to come by. They're presumably more expensive than the more basic ectomycorrhizal fungi that don't have that capacity. It's been demonstrated quite nicely in recent papers in phytology. So you look at a nitrogen gradient and they've got composition of the ectomycorrhizal fungal community changes, a lot of turnover. And it's the genes for organic nitrogen extraction are concentrated. They're much more common at the nitrogen pool end of the gradient.
In the fungi. So it's a facultative, like the plant can.
Plants
So, yeah.
plant can pick and choose. Yeah. I'll have you thanks here today. Yeah. Yes. That doesn't look like a happy face.
All right, control my facial expressions. Thanks, Chris. Yeah, I guess I've just got a couple of specific questions around the growth rate, light availability graphs you were showing. So on one of them, I think it was the Reore where you show that there's yeah, that one. So blue is
Broadleaf forest.
Broadleaf forest. It looks to me like that is a rather non-linear relationship. I mean, potentially, yeah, I'm just wondering if using a piecewise regression or something like that would actually show that it just looks like a threshold response there where, you know, below the light levels that you get in beech forest, it's very minimal growth rate and then it just takes off. I mean, with some pretty messy data, but it just seems to take off in the same form that you get in the beech forest where light availability is higher. I'm just wondering if.
Did you notice this point up here?
Well, yeah, I'm kind of ignoring that one, but just one point. Yeah, I'm just wondering if there is, yeah, perhaps just a bit of a threshold response there that.
I'm not sure I've ever seen a plant respond that way to light.
Which you say you got these from a nursery.
Some of the seeds. We grew some of them ourselves. Yeah. And we got I think 4 of the species. We went down and we got little seedlings and put them put them under the mist for a while, and then put them in they call those root trainers, right? And they grew nicely. Then we got damping off some of them, and so it affected two of the species, I think Hino and whatever.
Okay.
And we had to buy two of the species from nurseries because we couldn't find any other stores around Lemonwood. And then I ordered beech too because I wasn't confident we'd have enough beech seedlings. And I wish I'd just planted my own beech seedlings, which is good because the seedlings the nursery sold us were crap. In contrast to Lemonwood. And I should have just thrown it out.
I mean, it's great doing experiments. I think results are pretty good. Considered knowing how experiments can go. But I just wondered, like, I know Stephen Hubble in neutral theory, his monograph, he talked about Panamanian forests with certain species where shade tolerance across, well, you're within a species, the intraspecific variation was, really wide as what they saw in the specific variation. So I just wonder how much phenotypic variation there might be, and that potentially adds a bit of noise to what you're hoping to achieve here. Yeah, I mean, you're probably aware of this already. I don't want to point out obvious.
I was probably trying not to think about it.
Okay, yeah. Yeah, I don't know if there's some way you can quantify that.
Not at this stage.
No.
The seedlings don't come with their own mycorrhizal.
The ones that we plucked out the forest properly. And
Nursery ones are usually infected with sort of cosmopolitan species.
well, whatever they did in the nursery, they did a good job with the lemon the tortoise, but not with like stick the lemon and the tortoise on the ground and start sprouting leaves. Practically. Good material.
You have a lot of mortality, different treatments, different species, or just wonder some sort of survival analysis models that you use for showing the different stands that impacts you.
I'm not sure we've got enough data to do it. You big samples for survival analysis, and I think our sample size kind of. I mean, they're not even ideal for studying growth.
Yeah.
So, but we some species were more affected than others. Mortality. Basically, we got mortality in just about everything except children. Maybe we lost one or two children. Yeah, a couple of times.
Just one more question about the same one, but for beech. You had the strange result that beech seems to be growing better
You're in the
non-beech forests. But I mean, would that just be because they're competing against AMF species, right? And if you plop them into the broadleaf forest, they get the advantage of shortcutting the nitrogen cycle, but otherwise they're competing against other beech that are doing exactly the same thing, and competition is just stronger. Specific competition is higher.
yeah.
I mean, if that's a significant result, even. But
Beech would just invade rainforests.
eventually, I mean, I don't know. Like, it might take a really long time. But are these patches
There would disperse
growing out, or
beech.
They're pretty remarkably sort of sharp through most of these patches. Remarkably sharp.
But I mean, when they're regenerating in, like tussock land or something, they just basically grow out like sort of one row at a time. Would they do a similar thing?
I think
Matrix?
Matt McGlone's written about this, that expansion of beech in tussock is much faster than in forest.
The old explanation is they just don't have the ecto species present in the soil
But they would if they're in a hatch, right? They haven't
on the
there.
margins, but if they disperse into a stand,
Right.
that explanation is still used, that beech can't disperse because they don't have the
But surely they have above ground fruiting bodies. Surely the seedlings. Surely the spores would get around. Yeah.
But do you know if you looked into whether or not these patches seem to be like just continuously expanding and eventually a disturbance might knock them back or something?
It might be worth looking at the ecotomes there. Maybe there's a little thesis topic there for somebody. Summer scholarship project. I was thinking of doing that actually I'm going in shortly to a couple of months to the other site I showed you, Rohini Corner. And initially I thought about trying to measure, kind of use aerial photos actually to measure the rate of expansion, but then I decided it probably wasn't feasible, because what we'll see mostly is just the growth of the crown, tree crowns from on aerial photos. You'd have to do it on the ground like you suggested.
Trees, I guess. When you need to do that, I mean, presumably it wouldn't be that fast, right? Take a few years to see.
Well, the stands at Ruhini Corner, they all seem to be, it look like they're relatively young. And I think somebody got some age data out of stands, and none of the beech trees are very old. They all look like they're about the same age. So there's no huge beech trees in the stands. So you would see, if you saw any expansion of those patches, they're just little patches, on a sequence of aerial photos or satellite photos, you'd probably be seeing ground expansion. Because there's no monster trees in there. There's nothing bigger than about this. How they got in there?
It's black beech, not mountain.
It's black beech here, it's mountain beech at Ruhini Corner.
Oh, is it? Okay.
Yeah. There's red beech. If you go further west, there's red beech stands as well, coming in from the other side. So it was Jeff Rogers who did some nice work on Ruhini Corner back in the 80s.
Just looks cool.
Yeah. It's a long walk in there if you're going to go for it.
I've got one last question, and it's a bit out there, but I've always wondered about the beach gap in the South Island. I'm wondering if your research is.
I think it just ended. Oh no, it'll still be.
Yeah, I wonder if your research would figure in and explain. Well, there's obviously other hypotheses around why the beach gap is there.
Seems still there.
I'm interested in
Yes,
knowing
we
if
still do.
research might be.
Absolutely. Yeah.
Okay.
Yeah. So
To consider that problem.
sure. Beech appears to be slowly invading on both ends of the western beach.
Yeah.
It's very slow. So that conifer-broadleaf forest is pretty resilient as a in the case of Taranaki, it's just no beach within the so I don't know how beach is going to play there. So, Jude Island is another beach cabinet, but hard for beach to cross the strait.
Yeah, okay.
But we know it's a right to find beech forest growing in a climate that's changing. It's presumably it's been wiped out by the ice, and it just didn't disperse fast enough to get in before the other trees. Yes, probably. Yes.
So, I'd like to welcome any additional questions to kind of come along with us. There are some people waiting to enter the room, and I'd like to also thank you very much for